So we're going to talk about molecular and genetic pathways in lymphoma, and the prototype disease, if you want to address this issue, because I was thinking about it quite extensively when I got the title, is to discuss diffuse large B-cell lymphoma. And the reason is because diffuse large B-cell lymphoma is the most common of the non-Hodgkin's lymphoma. It is more than a third of the cases that we see in our practice. The other thing is that it's been extensively investigated in terms of molecular biology. There's been a lot of publications regarding its biology and the genetics of diffuse large B-cell lymphoma. And the other thing is, over the past decade, we've had many phase 3 clinical trials in diffuse large B-cell lymphoma. So we will retrospectively look at these things by reviewing the pathophysiology of diffuse large B-cell lymphoma, the expected outcome of current treatment for this disease. We will review the important clinical trials with targeted agents in diffuse large B-cell lymphoma, and finally the progress in the classification of diffuse large B-cell lymphoma and how we can improve clinical trial design and outcomes in the era of precision oncology. Now, we all know that diffuse large B-cell lymphoma is a heterogeneous disease in terms of morphology, phenotype, molecular genetics, and also clinical features. And we all are aware of the landmark study that was published in 2002 by the French group, in which patients were randomized to CHOP or CHOP plus rituximab. And this landmark study showed that the addition of the monoclonal antibody improves progression-free, but most importantly, overall survival in these patients. And as you can see here, these are the updated 10-year figures that were published in 2010. There's a significant, this significant difference is maintained, as you can see. 10 years progression-free survival, 36% versus 20%, and 44% versus 28% overall survival. This was, of course, confirmed in the younger patients in other trials. And this study established R-CHOP as the gold standard treatment for diffuse large B-cell lymphoma. This stands up to date. And over the past 25 years, we've relied on the International Prognostic Index to risk stratify our patients. And what we can see here that it relies basically on clinical factors, and the biology is strikingly not available in these prognostic markers, as you can see. But in 2000, there was a very important publication by the NIH group under the leadership of Lou Stout, with the use of cutting-edge technology at that time, which was gene expression profiling. They demonstrated that there are two distinct molecular subtypes of diffuse large B-cell lymphoma, varying between them in the expression of thousands of genes. But if you take out the noise, what you can see is that you have gene expression profile that mimics that of the germinal center, and another gene expression profile that mimics that of the activated B-cell. So we have two distinct types. There was another type, of course, which was unclassified, which has been an issue of debate, and we'll see further on if there's been any progress in these patients too. So what was the value of this gene expression signatures in the era of R-CHOP? The answer is there was, in the CHOP era, if you look at the survival curves, the prognostic significance of GCB versus ABC applies. But even in the R-CHOP era, although the addition of rituximab improves the survival in both subgroups, again there's a difference in favor of better outcome in patients who have the GCB subtype. Now, because this technology was not available and is still not available in practice, a more easily practical approach to define these subtypes was defined by using immunohistochemical classifier, and the Hans algorithm, which relies on three markers, the CD10, BCL6, and MUM1, has a concordance of 80% in separating the GCB from the ABC, the non-GCB subtype. There have been other algorithms that improved up to 95% of this concordance. So with these algorithms in daily practice using immunohistochemistry, we are capable of distinguishing GCB from non-GCB subtypes of diffuse large B-cell lymphoma. So based on this, it became evident that it was interesting to do studies in patients who have GCB versus non-GCB subtype. So in the WHO 2016 classification, it became mandatory to be distinguished by the histopathologist if the patient's pathology is of germinal center origin or of ABC origin, and it was also requested to define if they have co-expression of MYC and BCL2 at the immunohistochemical level, because we know that these patients do worse than those who are not double expressors. And it was written in this paper published in Blood that the mutational landscape will be better understood, and to see if this makes has any difference in the clinical has any impact on the clinical outcome of these patients was to be determined. Now, of course, next generation technologies refined our knowledge of B-cell lymphoid neoplasms, and especially diffuse large B-cell lymphoma. And what we did, and what we found out by these studies is that the ABC subtype relies on chronic active B-cell receptor signaling. So there's continuous signaling from the B-cell receptor, and the downstream pathway which leads to NF-kappa B is continuously downstream signaling active. And what was found was that mutations in the downstream pathway are responsible for this chronic active B-cell signaling. For example, we have mutations. Can you see this? Or no? Oh, yes, you can. We have mutations in the item of the CD79A and B molecule. We have mutations in the CARD11 gene, and also mutations in A20, which is a part of the NF-kappa B pathway reverse signaling. And also what was found was that 30% of patients with ABC diffuse large B-cell lymphoma have a mutation in the MYD88, which is a part of the signaling of the toll-like receptor. And this leads to direct activation of the transcription factors involved in the NF-kappa B pathway. So we have two types of signaling. We have chronic active BCR signaling, which relies on multiple downstream pathways and is associated with ABC diffuse large B-cell lymphoma. And we have another form of signaling called tonic BCR signaling, which is antigen independent and is mainly seen in Burkitt's lymphoma and GCB type diffuse large B-cell lymphoma. Now, as you can understand, this led to the development of molecules that target downstream gene signals of these pathways, as you can see here. And it was evident, it was as a consequence, these molecules were tested in relapsed refractory setting of diffuse large B-cell lymphoma. For example, this is a slide I dug up, I had in my archive from 2012, when the BTK kinase inhibitor ibrutinib was tested in relapsed refractory diffuse large B-cell lymphoma. And as you can see here, there was a 40% response rate in patients of the ABC subtype compared to 5% in the GCB subtype. So this led to a phase 1 study combining ibrutinib with R-CHOP, and it's a busy slide, but simply it tells us that you can combine ibrutinib with R-CHOP without any serious toxicity, which was the beginning to set up a phase 3 trial comparing ibrutinib R-CHOP with R-CHOP. It was also demonstrated that lenalidomide as a single agent has better activity in the non-GCB subtype. Sorry, I can't use this. Anyway, so as you can see here in the slide, you have a significant 50% response rate compared to 10% in favor of patients with non-GCB, and a better progression-free survival in these patients, suggesting that lenalidomide has a selective, better activity in patients with the non-GCB subtype. So if we look at the recently reported phase 3 trials in first-line diffuse large B-cell lymphoma, as you can see here, if we start first of all with the dose-adjusted R-epoch, which was heavily invested by the NCI group, compared to R-CHOP, disappointing results. It was a negative trial. There was no difference in event-free survival at 5 years between these two arms. Again, looking at a new antibody, a new anti-CD antibody, the obinutuzumab. So obinutuzumab-CHOP versus R-CHOP. Again, this was published recently. No difference in progression-free survival, a negative trial. Looking at various targeted agents such as azacitidine, that was or placebo maintenance in patients who had received R-CHOP. Again, negative trial, no difference in progression-free survival. Everolimus maintenance or placebo, which is an mTOR inhibitor. Again, no difference in disease-free survival at 2 years. The REMARK study, which was published recently, and it was looking at maintenance lenalidomide for 2 years in patients aged between 60 and 80. This was a positive study in terms of progression-free survival, but as you can understand, it had excessive toxicity in the lenalidomide arm, if you consider the age of the patients. So this is a half-full glass, let's say, of water, and although it improves progression-free survival, there was no difference in overall survival. And the main trial, which looked at the VEGF inhibitor, bevacizumab, with or without R-CHOP, again, a negative trial. This was actually terminated early because of excessive toxicity in the bevacizumab arm. So these phase 3 trials were quite disappointing. Now, we discussed about gene expression profiling, okay, using the immunohistochemical cell of origin algorithm. But technology now has improved, and what you can do is you can take formalin-fixed, paraffin-embedded tissue, and you can use gene expression profiling in very small kits that are available. And these types of technologies, one of them is the nanostring technology. The other is the Modiplex. These are companies that have used this technology and created small chips where you can actually very efficiently and quickly determine with gene expression profiling the cell of origin. And this may replace immunohistochemistry in the future. So we shifted from the International Prognostic Index to gene expression profiling. From there, for reasons of simplicity, we shifted to the immunohistochemical algorithms, like the Hans algorithm. But now we have these very nice fancy machines with these platforms that can very easily and efficiently and rapidly and consistently report on the cell of origin of these tissue samples. So there have been studies looking into this. These are phase 3 trials, again in first-line diffuse large B-cell lymphoma with cell of origin determined by either gene expression profiling, nanostring technology, and there is also 2 studies here by using Hans algorithm. So the Remodel B study looked at bortezomib plus R-CHOP versus R-CHOP, the first study. This used gene expression profiling to separate these 2 entities. And as we know, bortezomib targets the NF-kappa B pathway, and again, negative trial, disappointing results. Progression-free survival at 30 months did not differ. The Pyramid trial again looking at bortezomib plus R-CHOP versus R-CHOP in the non-GCB group. Again, negative trial, very disappointing results. The robust, which has not been published yet, this is looking at lenalidomide plus rituximab, that's R-squared CHOP versus R-CHOP, and this is using nanostring technology and looking at if there's a difference in the ABC subtype. The only results we have are on the nanostring gene expression profiling feasibility, which was reported at ASCO this year, demonstrating that you can actually have results within 48 hours and stratify these patients into the study. The last and not least study is the rituximab ibrutinib CHOP versus R-CHOP study. This will be presented at ASH this year. This is a negative trial, and the results were not available, but there was a press release by the parent company that said that this is a negative trial and it did not meet its endpoint. So we have numerous attempts to improve on R-CHOP that have been unsuccessful. And what is striking is that some of these studies, for example, the Pyramid trial, showed 2-year progression-free survival of 78% for non-GCB. This is unrealistic in the real world. We see we see progression-free survival 2-3 years of 30 and 40% in historic controls, which means that there is a terrible selection bias in these studies. So what they do is they exclude real-world patients. If you're not fit, you're not going to get onto these trials, okay? So all these exclusion criteria actually exclude the real-world patients, and this can have a wash-away effect on the phase 3 and phase 2 trials. That's why we're not getting any positive results from these trials. The other thing is that diffuse large B-cell lymphoma is a vastly heterogeneous disease, well beyond the cell of origin that we know, the GCB, non-GCB. And recent studies have identified subtypes that cross these boundaries of the cell of origin classification. So what we need now is we need new coordinate genetic signatures with specific targeted genes that should be that we can target if we want to move closer to precision oncology. So, all these so the NCI group with Lou Stout and also the Boston group, they went back to the drawing board to see what's going on. Why don't things work out as they should? So the Lou Stout group published recently in the New England Journal of Medicine a article in which they took samples from 574 patients with primary diffuse large B-cell lymphoma. They did exon and transcriptome sequencing, array-based DNA copy number analysis, and they did also targeted amplicon sequencing of these 372 genes that they found. And what they did is they tried to group these genetic alterations into subgroups, and what they found was they found four distinct genetic subtypes of diffuse large B-cell lymphoma, which accounted for about 50% of their cases. So the other 50% could not be classified. But still so, that is some progress. The first subtype is the MCD subtype, and what they found is that they have a double mutation. They have the CD79B I showed you earlier, and the MYD88 mutation. This subtype has extensive extranodal involvement. Okay, it also has an immune editing genotype. The vast majority of patients, as you can see here, are of ABC subtype, and the pathogenesis is related to primary extranodal lymphoma. So these are a type of primary extranodal lymphoma, and what they suggested is that we could target this distinct subtype, this distinct subtype with distinct signature, excuse me, with BCR toll-like receptor signaling pathway. For example, we can use ibrutinib or lenalidomide in these patients. I'll remind you that we mentioned earlier that MYD88 mutation is evident in about 30% of patients with ABC, diffuse large B-cell lymphoma. Now, the term synthetic lethality corresponds to what? It means you have a specific cell that survives, but if you expose it to a specific drug, nothing happens to it, doesn't die. But if this cell has a specific mutation, the drug that didn't do anything to it before, if it acquires this mutation, it kills the cell. So what am I trying to say? So if you take ABC, diffuse large B-cell cell lines, and you add lenalidomide to these, you see a lot of these patients, a lot of these cells die. This is because there is production of interferon beta. Now, if we go back to this pathway, the toll-like receptor pathway, lenalidomide blocks IRF4. IRF4 is interferon related factor 4. And what it does is it represses production of interferon beta. So IRF4 represses production of interferon beta. If you block it with lenalidomide, you have overexpression of interferon beta. Interferon beta kills lymphoma cells which have the MYD88 mutation. So, what could we do in this specific subgroup of patients? We could use lenalidomide to block IRF4 and kill the cells, but also we can block IRF4 with another molecule, ibrutinib, which is a BTK inhibitor, so we can have dual blockage of IRF4. This was done in knockout in mice in the lab, and it showed that you have excessive production of interferon beta, and these cell lines die. So this might actually work in this subtype of patients. The other subtype that was found was the N1. This is dominated by Notch1 mutations. It's of predominantly ABC subtype, as you can see here, 95%. And they suggest that with Notch or immune checkpoint inhibitors, you might be able to get better outcome in these patients. The other subtype is the EZB subtype. This is characterized by BCL2 translocation, EZH mutation, and JAK-STAT upregulation, and it's predominantly, as you can see here, of the GCB subtype. And what they propose is targeted therapy with BCL2 inhibitors such as venetoclax, the epigenetic modifier EZH2, and JAK-STAT pathway inhibitory molecules. If we go to EZH2 is a epigenetic gatekeeper of the germinal center. And what EZH2 does is, although it's repressed in the naive B cell, when it enters the germinal center, it's overexpressed, and what it does is it helps proliferation of the B cells in the dark zone of the germinal center. But normally, when the cells exit the dark zone towards the light zone, and this is because they terminally differentiate towards plasma cells, and we have upregulation of IRF4 and NF-kappa B, EZH2 levels drop. But if you have a mutation of the EZH2, what happens is you have continuous proliferation of the, you have hyperplasia of the germinal center, and acquisition of other oncogene events such as BCL2 translocations lead to the development of GCB, diffuse large B-cell lymphoma. Also, EZH2 can also be upregulated without mutation, but by other oncogenic hits, which again will lead to GCB, diffuse large B-cell lymphoma. And EZH2 mutations are identified in 20 to 25% of patients with diffuse large B-cell lymphoma and of the GCB subtype. Now, if you use EZH2 inhibitors such as tazemetostat, and if you combine them with BCL2 inhibitors, or even with hypomethylating agents such as 5-AZA, you can kill these cells very effectively. And this has been demonstrated in follicular lymphoma with the EZH2 inhibitor tazemetostat, as you can see here, in patients who have follicular lymphoma with mutated EZH2, the response rate is 92% compared to 26% in patients who have wild-type EZH2. So this is a very promising agent. And the last subtype is BN2, in which you have mutations of Notch2 and BCL6. These are, interestingly, 40% of this subtype are from the unclassified gene expression profiling category. The other 40% is from the ABC, and the other 20% is of GCB origin. And it's been proposed that these might have marginal zone lymphoma origin. And a variety of targets, as you can see here, I have to close my talk, could be used in these agents. Now, just briefly to summarize, EZB2 and BN2 subtypes, they have better progression-free survival and overall survival. And if we look at ABC, diffuse large B-cell lymphoma, those that are of the BN2 subtype or other ABCs have better outcome than MCD and N1. And in terms of GCB, diffuse large B-cell lymphoma, patients who have the EZB subtype have poorer prognosis compared to those of other GCB. Now, another group from Boston did a similar study, and they found 5 robust diffuse large B-cell lymphoma subtypes. They called them C1, C2, C3, C4, C5. We don't have time to go through them, but just to summarize, we're going towards an approach where we're getting more complex classification of diffuse large B-cell lymphoma. We're looking into specific genetic signatures which correspond to specific targets. That is the way we will go forward. Otherwise, we will be stuck to R-CHOP for the next years. So these studies, as you can understand, will take a lot of time, okay? So the take-home message, the promise of targeted therapy in diffuse large B-cell lymphoma has not been fulfilled during the past decade, okay? But the premise of targeted therapy is being fulfilled. We have next-generation technologies that are improving our understanding of the genetic landscape. We have novel classification of diffuse large B-cell lymphoma. I gave you a hint with a new classification we're shifting towards. But the complexity of the disease underscores, as I wrote here, right here, the need for precise patient stratification in order to identify those who are most likely to respond to targeted therapeutic approaches. Thank you.